My pleasure to introduce our second speaker, Floris Engelhardt from Kano. And we'll be covering a variety of platforms, moving away from AAV.
Yeah. I recently came across this video. It's like 20 years old, from a comedian that was talking about religion that had me think about DNA. And the reason is that he was dramatically annoyed about the conversation about messengers and delivery in religion, saying like, okay, everyone is fighting about the messenger, but did you get the message is actually what we want to think about. And so I want to talk about DNA as the message, as the payload that we use in these therapies. And I think one of the big questions here that we've realized recently is, what can the payload do for the delivery problem or for the delivery solution? And when we think about viral vectors, AAV has a single-stranded DNA genome. And so the payload there already contributes with its structure and function to the delivery aspect and the efficacy. But we've also seen that there are a couple issues that we see now with AAV or other older generations coming up. And so a lot of these people are moving towards non-viral delivery, where then the question becomes, if I deliver a DNA, then I face DNA toxicity because double-stranded DNA is picked up by the immune system. And the third part that we also see is when we then move to lipid nanoparticles is the question of how do I get from the cytosol actually into the cell nucleus, because suddenly we enter into a new delivery problem of nuclear limitations. And so what we've recently seen is that there is an opportunity to switch from a double-stranded DNA that everyone currently thinks about to a circular single-stranded DNA as a DNA template. And that's what we do at Kano Therapeutics. One thing that I want to show you here is the reason why we even started this. And that is, if you think about the immune system as a pattern recognition system, a very common pathway is the cGAS-STING pathway. And as you can see here, when you think about phosphorylation of STING, turning on that pathway, double-stranded DNA is just really, really triggering that pathway. And it doesn't need to be circular, it doesn't need to be plasmid, it's just the double-stranded DNA pattern that triggers that pathway. Single-stranded DNA now does not, and that has a strong impact on interferon secretion, which then in return has a strong impact on the DNA toxicity and the dosing that you can do. But there are more advantages too. If you think about single-stranded DNA, from an engineering perspective, it's half the weight of the material with the same amount of genetic information. And when we design it as a fully circle, then it doesn't have any open ends, which makes it more stable than any linear DNA. But also it gives us a unique opportunity to think about locally structured features that are self-folding that allows us to now consider the DNA as a functional part of our delivery tool. And so if we think about single-stranded DNA at Kano, we need to first take a step back and say, in reality, this is not going to be a fully single-stranded piece of genetic information. It will fold back on itself. We will have collapsed transient features, but we can use those. We can consider long-range interactions, for example, across the thousands of nucleotides that we're working with, or we can design discrete double-stranded features that then allow us to bind proteins. So it's really the idea of making the payload a functional part and considering, can we do editing tool binding? Can we design immune stimulation features? CPG islands, yes or no? What do you want to have as functional part of your delivery tool? And so in our case, we don't have our own assets. We really work with our partners to think through what is your asset that you want to bring to the clinic. Our current focus, or where we see a lot of 50% of our clients are going for, is obviously the CAR T field. One thing that, first data I want to show you here is from a BCMA CAR T cell therapy. It's an ex vivo non-viral therapy where the idea was to say we want to create a knock-in to the track locus, so targeted site-specific insertion using homology-directed repair. But the partner wanted to know how we compare to other DNA forms. In this regard, as you can see here, the circular single-stranded DNA shows higher knock-in efficiencies or more edited cells than a linear single-stranded DNA or a linear double-stranded DNA, which convinced our partner for us to be the best path forward in this specific therapeutic approach. But we were also thinking about, in this regard, this is a BCMA. So you could package that into an AAV. What if you create a payload that you cannot package anymore into an AAV? Because that's when knock-in efficiencies actually really drop. But in our case, what we can design is something that even at larger sizes still gives you high insertion rates with a standard insertion construct. We're now looking to work with new partners, data I can show you, that even goes beyond CRISPR and goes towards recombinases that would then allow you to go into non-viral, non-dividing cell therapies. And the last thing I want to show you here on the right side is really the idea of going in vivo. Because now we have a huge field of people who have developed lipid nanoparticles or other encapsulation technologies for mRNA. But what if we package a DNA into that nanoparticle, and we can allow us to deliver it into humans? And as you can see here, one of our partners had the question of what happens if I package a single-stranded DNA instead of my double-stranded DNA into my lipid nanoparticles. This is a mouse study, and you can really see how this high inflammation signal that is caused by DNA toxicity is related to the double-stranded DNA, whereas if you switch to a single-stranded DNA, you have a very benign form, and you can dose higher and think about like, what is the next step for my in vivo therapy? Now the question is, if circular single-stranded DNA is such a big thing, like why didn't people do that like 5 to 10 years ago? And so when we started Kano about 4 years ago, we looked at the market, and making long single-stranded DNA at large scale was simply not possible because chemical synthesis doesn't do it, and enzymatic processes are too expensive. So what we were coming up is, can we create a cell-based process that can use all the infrastructure that plasmid DNA already has, but make a single-stranded DNA? For 2,000 to 14,000 nucleotides, the largest genetic construct we did was actually a dystrophin transgene so far. We can make it cost-efficient and scalable. We allow that to be a production E. coli-based process with a secreted capsid, where we then purify out the single-stranded DNA and continuously can grow that process. It allows us to really scale and make cost-efficient large amounts of single-stranded DNA, thereby now unlocking the whole field for these new type of genetic template. And so with all that together, we now see the opportunity of manufacturing and design coming together for the DNA payload. And so what we do right now is that we have looked at where can we best place this technology most efficiently. And we are already working with 4 public pharma companies and a couple of private biotechs across different indication areas, really allowing us to diversify the different parts and thereby create a community of people that all face the same purpose by also using several different modalities and technologies: pancreatic cancer, blood disorders, solid tumors, liver protein loss. So when we look at this, is really for us the opportunity to start with every one of our partners early when they think about lead optimization. And we really look for drug developers that are in the ex vivo and in vivo CAR T field, have novel gene delivery tools, and really want to work with us to think about the cargo. But really like the idea of like, you have an asset in mind, you have your first technology that you have there, now think about what is the perfect DNA message that you want to send into humans. And that's when you come to us. Thank you very much, and I'm happy to talk later.